also a movie here from Gavin Schalk and Nick Anderson and Eric Luthard using ECoG recordings here. You see the cables coming out of these patients going to the amplifier. They are connected to the PCI system. And the patient is moving his hand to move this item around, and he's trying to fight against the other items. So I guess everybody knows the game. It's quite old, but it shows the principle how you can use motor movements for controlling PCI systems. And if you have calibrated your PCI system on the real movements, you can also switch to the imagination of movements, which is shown here. And you get basically the same results. So now you see he's still moving the item around, but he's not moving his hand. Yeah, there are also some clinical risks associated with ECoG recordings. Here are some slides from Eric Luthard from St. Louis. If you place the EEG electrodes on top of the head, you can maybe abrade the skin a little bit, so there are just minimal clinical risks associated with that. If you place the ECoG recordings epidural, then you have only a little risk for destroying the cortical surface. But the biggest disadvantage is that the electrode grid can move related to the brain surface. So if your brain is moving around, you are picking up different activity depending on the location. If you put the ECoG grid under the dura mater subdural, you have the big advantage that the brain and the ECoG grid are quite tough together, so you are picking up always the same activity, but the clinical risk is a little bit higher to produce, for example, bleeding. If you look at single units, then of course you have this very fast spike trains coming in that you can analyze. But for the patient itself, it's more risky than the ECoG recording, and some patients can also get epilepsy because of the implants. So it's not that easy to do. So we used all the technology that I explained up to now for real-time position reconstruction with hippocampal clay cells. So there was already previous work done in this field, and they looked at narrow tracks, at circular environments, and rectangular environments. So Chang, for example, had a figure 8 maze where the rat was running around, and he reconstructed the position inside. He looked at different algorithms, for example, Bayesian and template matching algorithms, and found out that basically Bayesian algorithms work best for this task. And Jensen and Lisman had a linear track with 2 meters length, so the rat was running around inside this linear maze, and they improved the classification by looking also at data activity, because that's also influencing the spiking, and they removed segments where they didn't find spikes. So if a rat, for example, is not moving around, the number of spikes is reduced. And of course the position reconstruction accuracy can be improved if you remove these intervals. And they noticed also if the rat is at the food stand and eating, then they get a lot of artifacts. So if you remove these sections out of the data, you can also improve accuracy. Then Brown and Barbieri used circular environments with 70 centimeter diameters, so they had, for example, 44 neurons for doing that, the tracking error of 5 centimeters. And Wilson had a large rectangular environment. He used 76 cells and reached about 9 centimeters accuracy. So the goal of our study was to look at different rectangular environments from 0.5 meter up to 1 meter, and to use the same algorithms that were shown beforehand to work quite well, but we wanted to do everything in real time. So here you can see the principle of position reconstruction. Step 1, we acquire the data. So we have the dead roads, the spikes are coming in via the biosignal amplifier, we record them. We have a video tracking system so that we know where the rat is moving around. It gives us the X and the Y position. And this is step number 2, we are encoding in the encoding step, we do manual spike sorting. This means we load all the spike waveforms and we sort them. This means each spike gets a neuron ID assigned. So at the end we will know how many neurons we had, and each spike corresponds to one certain neuron. If we have done this, we are able to calculate the firing maps, to identify the place cells, and to know if we have nice place cells. So this guy, for example, is only firing in the upper part of this rectangular environment. And then we can also calculate spike templates by just averaging all the spikes of one particular neuron. And from the position information, we can calculate the probability distribution where the red was mostly located. And then we go to the real-time decoding step. We record again the spikes. We automatically sort now the spikes. So the spike is coming in. We say it was neuron 1, 2, 3, and so on. Then we calculate the firing rate and put it into the Bayesian two-step algorithm to predict the position. And finally, we compare with the position coming from the video tracking system to get the accuracy. So here you see the video tracking system. You need a video camera on a tripod mounted above the arena where the rat is running around. It's connected with a frame grabber to the notebook and gives you excellent point position. Then we also record the spikes. So here you see a typical trace of spikes coming in. This here is just a noise. It's about plus minus 30 microvolts in this case, and the sharp high peaks are the spikes of one tetrode. So the different colors show the different channels. In this case we have 4 channels because it's a tetrode, and the goal is now to sort them. So for the offline position reconstruction, like mentioned before, first we do the manual spike sorting. So we look at the amplitudes of the spikes, at the shapes and at the timing, and we remove all the spikes which are too close together, and for that we use the inter-spike interval histogram. On the next slide I will show you how this works. And finally we calculate the firing rate maps. Therefore the arena is divided in certain pins, 64 by 64 pins, and each pin gets a certain ID, and then we calculate the firing rate and by just summing up all the different spikes, and this allows us to have the firing fields. So if we have here a time series coming in, so the vertical line indicates the different spikes, and we see that the time distance between the different spikes is different. So we can make a histogram, having here the inter-spike interval, and here we count how many intervals are the same. And spikes have a refractory period. This means they cannot appear within 1 millisecond. So if here we would see some activity, we would remove all these spikes from further analysis because it would be an artifact. For the manual spike sorting, we are using the signals recorded with tetrodes, and tetrodes have a very nice feature because you have 4 channels, and let's assume there is one neuron located like the green dot here. Then of course on channel number 4 the amplitude would be a little bit higher than on the other electrodes because it's closer. So we can plot the activity of the different channels against each other. So if we have, for example, channel number 1 and channel number 4, then we would have a very high activity on channel number 4 because it's so close, and a very low activity on channel number 1. And we can do this for all the spikes, and finally we will see that the clusters are forming up, and we just have to identify the clusters to find the different neurons. So in this case, for example, we would have 2 different neurons. This can be done manually, but it's time consuming, and sometimes it's really complicated, but there are also automatic spike sorting algorithms available. Yeah, we have also a cutting tool which allows you to do that. And here's a real recording from place cells, and in reality, as you can see, the clusters are overlapping. And this makes the spike sorting a little bit more complicated. Just checking. So we started a little bit later. Okay. So here, now from one recording, you see 8 different firing fields. This means for this single red, we had 8 place cells identified. And as you can see, the quality of the place cells is not always the same. And there are specific algorithms available which allow you to quantify how well this place cell is behaving. So we have, for example, the spatial selectivity, spatial information in bits per second, or spatial information in bits per spike. They were defined some years ago by SCAPs and are quite frequently used for identifying the quality of place cells. You can see this place cell, for example, is containing a lot of information, and it's very important for the reconstruction procedure. If you look at this place cell, for example, it's not very distinct. It has a very low bits per spike rate, and it will not help a lot for the position reconstruction experiment. So it's very important that you find good place cells to be able to do this. Then for the real-time position reconstruction, it was also important to have a real-time spike sorting algorithm available, which is performing as good as the manual spike sorting. So what we did is we calculated the firing fields for the manual sorting, the 8 cells again. Then we used this 3 sigma method. I will explain it a little bit later on how it's working. So we recut automatically the cells again, and you can see that the firing fields are matching very well. So it's a quality measure of our 3 sigma real-time spike sorting algorithm. Here we have the Pearson correlation coefficient, and you can see that mostly the firing fields are highly correlated above 0. So for the position reconstruction, after spike sorting, we put the spikes into a Bayesian 2-step algorithm, which was developed by Chang some years ago, which has a continued constraint. This means we are looking also at the velocity and the previous predicted position of this animal, because it's impossible for the rat, for example, to go from one corner of the environment to the other corner within one sample step. This helps to increase the performance of the algorithm. Then we have a time window sliding over the whole data set, and within each time window we calculate the firing rate of each neuron and compare to the training data. And this gives us the position where the rat is located at this certain time point. And the reconstruction algorithm can be improved by several parameters. For example, the length of the reconstruction window. So if the window is too small, you don't have enough spikes, of course. If the window is too long, like 10 seconds or 20 seconds, then it's not really the position that you're looking for, because the rat was already running around. So it must be optimal to have nice accuracies. You can also define the step size, how fast you move the sliding window over the data set. You can define the smoothing of the place cells, which is especially important if you just have a few place cells. The minimum number of spikes in each segment are important. If you don't have spikes, there's nothing what you can reconstruct. And the number of quality of place cells, of course, is important. So here you see a recording setup, a rectangular environment. The rat is moving around, connected to the main amplifier with this cable. There's one tracking PC on the left-hand side, the biosignal amplifier, and the spike sorting compute on the other side. This is not a result of one rat. In red we see the X and the Y position of the video tracking system. In blue we see the X and Y position that were reconstructed with the place cells. And you can see both follow each other quite well, but there are also sections where the error is quite high. And this is mostly related to sections where the rat was not moving or just standing, and we just get not enough spikes to do the spike sorting good enough.